Hi everyone. In the immunology subject, we are going to discuss another topic called major histocompatibility complex or simply MHC. In humans, this complex is going to be named as HeLa complex, that is HLA, human leukocyte antigens complex. Okay, suppose when a disease associated proteins occur in a cell, they are broken into pieces by the cell's proteolytic machinery, and these cell proteins become attached to the antigen fragments and transport them to the surface of cell where they are presented to the body's defense mechanism. These transport molecules are going to be termed as MHC proteins, and without these MHC proteins, the T cells cannot distinguish between the self antigens and non-self antigens. So what are these self antigens? The self antigens are also called as allo antigens. So according to Mr. George Snell, he said that MHC complex is a closely related genes or a cluster of genes that determine the transplantation acceptance or rejection between two individuals is going to be called as MHC complex, major histocompatibility complex. That means the cluster of genes which are going to determine whether the transplantation have to be accepted or rejected between two individuals is going to be called as MHC complex. And the MHC genes are going to give rise to or encoded proteins which are expressed on the cell surface, and these protein molecules are going to be called as MH antigen or MHC molecules, and simply we can also call them as H antigens. The full form of this MHC is going to have the different names in it. Histocompatibility means histo means going to be of a tissue name, and the compatibility means getting along, that means agreeable, acceptable. Okay, so that is compatibility. And the complex means the genes which are located or localized to a large genetic region containing multiple loci is going to be the complex. So that's how MHC, major histocompatibility complex term, is going to be of having the meaning. Now let's see the definition of MHC and what are the types of MHC in detail one by one. Now, MHC can be defined as a tightly linked cluster of genes, as a tightly linked cluster of genes whose products play important role in intercellular recognition and in discrimination, that means to distinguish between self antigens and the non-self antigen. Such type of the cluster of genes is going to be defined as MHC. Once again, the MHC can be defined as a tightly linked cluster of genes whose products play important role in intercellular recognition and also in discrimination between the self, that means to distinguish between self and non-self molecules. And in humans, the genes coding for MHC are present or found on the chromosome 6. Okay, and they are also going to be called as HeLa, that is human leukocyte antigens. And one more thing that we have to remember is on the chromosome 6, these MHC genes are going to be present on the short arm. So that's how the genes coding for MHC are present or found on the short arm of chromosome 6, which are also going to be called as human leukocyte antigen. So that's why here it has written as HeLa region, that is HLA, human leukocyte antigens. Okay, then moving to the types of MHC. As I told you, these MHC molecules are very important in the development of both humoral and cell mediated immunity. Let's see the types and then we'll move to the detail of functions, all those things one by one. So coming to the types of MHC. So as I told you, we are going to have the different types of MHC. Majorly, they are going to be of three categories. Okay, so here the HeLa region is going to have how many types of MHC? There are three regions or three types of MHC, major histocompatibility cells. Okay, so coming to the class 1. So here is a class 1. The class 1 molecules are located on three different loci, that is A loci, B and C. And these glycoproteins are expressed on all nucleated cells. That means the cells which possess the nucleus, on that we can find this MHC class 1. Okay. Then moving to the second type, that is class 2. So here is the class 2 molecules which are located on the region called as D region. So which are further having the different loci like DR, DQ and DP. And these glycoproteins are expressed on the cells called as antigen presenting cells, simply called as APC, which are going to process the not self protein. So those are going to be called as antigen processing or antigen presenting cells. So that's how the class 1 is going to be present or expressed on all nucleated cells, whereas the class 2 is mainly going to be expressed on APC, that is antigen presenting cells. Then there is third type of the class, that is class 3. Okay, so this is the class 3. The region or the area between the class 1 and class 2 is going to be the class 3. And these genes are going to code for complement proteins and cytokinins which take part in the immune system. But these class 3 molecules are not going to be expressed on any cell surfaces. So these are the three types of MHC. The class 1 is located in three different loci, A, B, C, and these glycoproteins are going to be expressed on all nucleated cells except RBC, which is a unucleated cell. Then coming to the class 2, molecules are located on the D region. This complete is the D region, but there are several different loci, that is DR, DQ and DP. And these are going to be expressed on APC, that is antigen presenting cells. And then the region between or the area between the class 1 and the class 2 is going to be the class 3, and these genes code for complement proteins and cytokinins, and these are not going to be expressed on the cell surfaces. So we will discuss all these three types in detail now, one by one. So this is what I have discussed in the figure. Okay, so let's come to the first one, that is MHC, major histocompatibility class 1. So here, this is a MHC molecule, okay, class 1. These are going to be expressed on all nucleated cells. That means the cells which are going to possess a nucleus. So you can have the expression of this MHC class 1 molecules on them. And they are present or found on plasma membrane of cells except RBC. So this is the plasma membrane, and that you can find this one. Class 1 molecules consist of two polypeptide chains. How many polypeptide chains? Two polypeptide chains. One is going to be the alpha chain. So this three pink color is going to be denoted as alpha chain, and the small one is going to be denoted as beta 2 microglobulin. So as it is having a very small size, this is going to be considered as microglobulin. Okay, so let's start with the alpha chain, then we'll move to the beta 2. The alpha chain is encoded in the MHC on chromosome 6. It has a molecular weight of about 45 kilodaltons. And this alpha chain is folded into three external domains. So this is one domain, 2 and 3. So you can see. So alpha 1, alpha 2 and alpha 3. So that's how the alpha chain is folded into three external domains which are termed as alpha 1, alpha 2 and alpha 3. And each of these domains are approximately of 90 amino acids long, and the alpha chain is inserted into the cell membrane. So here you can see the cell membrane by a transmembrane domain, which is of about 25 amino acids long, followed by a short stretch of hydrophilic amino acids which form a short cytoplasmic tail. So it is going to have a short cytoplasmic tail. Okay, so carboxylic terminal COOH. So as you can see here, this is a alpha 3 domain is attached to the plasma membrane by a transmembrane domain, and this alpha 3 is invariant, that is, it is going to be differ in different cells, and contains the binding site for the T cell co-receptor CD8. So remember, the alpha 3 domain is invariant and contains the binding site for the T cell receptor CD8, that is cluster differentiation 8. Whereas the alpha 1 and alpha 2 domains, which lie to the outside, forms the antigen binding cleft, or you can also call it as a pocket or a groove, and it is large enough to bind a peptide of about 8 to 10 amino acids. Now, once again have a quick review of this. So MHC class 1 is going to have the two types of polypeptide chains, that is alpha chain and beta chain. The alpha chain is encoded on chromosome 6 and its molecular weight is about 45 kilodaltons. And alpha chain is going to be folded into three external domains. This is alpha 1, alpha 2 and alpha 3. And the alpha 3 domain is invariant and contains a binding site for the T cell receptor CD8. Okay, so this is all about the alpha chain. Then moving to the beta 2 microglobulin, and this is going to be the second polypeptide molecule, and its molecular weight is only about 12 kilodaltons. It's very small. Okay, 12 kilodaltons, and encoded by a single gene on chromosome 15, whereas alpha gene is going to be coded on chromosome 6, and this beta 2 microglobulin is coded on chromosome 15, 1, 5, and it is bound non-covalently to the chain. Okay, it is bound non-covalent to the alpha chain, and it has been found essential for proper folding of alpha chain. Whether this have to have the proper folding or not, that is going to be decided by this beta chain. And it has no transmembrane region. Have you observed here, we don't have any transmembrane region. That means it does not penetrate through the plasma membrane. And this beta 2 microglobulin is going to be identical in all the cells, whereas the alpha 3 is invariant. Okay, so that's the main difference between these two. So that's all about the MHC class 1. So it possesses the two chains, that is alpha polypeptide chain and the beta 2 microglobulin polypeptide chain. Here we have three, and here is the one which do not possess any transmembrane. So then move to the second type of MHC class 2. So here these molecules are located on D region. So in the chromosome we have seen the HeLa regions, isn't it? So here you can see it is present on the D region. So up to now we have discussed the class 1. Now we are going to discuss this one, D region. And this are going to have the different loci. They are DR, DQ and DP. And these are going to be expressed on antigen presenting cells, where the class 1 is going to expressed on all nucleated cells, whereas the class 2 is going to be expressed on APC, that is antigen presenting cells. And now these antigen presenting cells are going to be categorized into two types, that is professional antigen presenting cells, and the second one is a non-professional antigen presenting cells. The professional antigen presenting cells are going to be expressed consecutively these MHC molecules. That means professional antigen presenting cells are able to constitutively express these MHC class 2 molecules. And these include B cells, macrophages and dendritic cells. So those are all going to be of professional APCs. And then coming to the non-professional APCs, which are going to be expressing the class 2 molecules for short period only, where it is going to have a continuous one, but here it is going to be expressed only for short period. And we can observe the cells included in this one are microglial cells of in brain and in fibroblast cells. So that's how the class 2 molecules are going to have very restricted nature. whereas the class 1 is going to have wide nature, but here it is going to have a very restricted one. Okay, so again, coming to the internal structure of MHC2, it is also going to possess the two polypeptide, that is alpha chain and the beta chain. Coming to the alpha chain, the alpha chain is going to have a molecular weight of about 33 kilodaltons, and it contains two external domains. So here you can see two external domains, that is alpha 1 and alpha 2, and it was penetrated or protruded into the plasma membrane, so by the transmembrane. Then coming to the beta chain, its molecular weight is 28 kilodaltons, and it contains beta 1 domain and beta 2 domain, and it also going to have the penetration into the plasma membrane. So whereas in the class 1 we don't have any what we call it as transmembrane region here for the beta 2, but here it is having a transmembrane region. And this beta 2 is the binding site for the T cell receptor, that is CD4. Whereas in the MHC1, that is major histocompatibility class 1, you are going to have the T cell receptor at alpha 3. But here in MHC2, you are going to have it as beta 2. So the beta 2 domain contains the binding site for the T cell co-receptor, that is CD4, whereas in the one it is CD8, that is cluster differentiation 8. Okay, then both the alpha and beta, that is here you can see the alpha 1 and the beta 1 domain, form the antigen binding pocket or a cleft or groove, whatever it may be, and it can bind the peptides containing of about 13 to 18 amino acids. So that means here is a region where we can observe the antigen binding pocket, and that is free, but in the class 1 it is going to be of a closed one. Okay, and it is alpha 1 and alpha 2, but here it is alpha 1 and beta 1, which is open. So these are all the differences where we'll discuss at last. Okay, so this is all about the MHC class 2, where mainly the importance of this class 2 comes as extracellular or exogenous antigens on cell surfaces. Then already I told you about the class 3, isn't it? The class 3 is mainly involved in of having the expression of complements as well as some sort of cytokinins, isn't it? Which take part in the immune system, and they are not present on any cell surfaces. That means they are not going to express on any cell surfaces, and those are going to be of MHC class 3. Okay, so now coming to the functions of MHC. So MHC class 1 and class 2 genes and their products provide a system for intercellular communication. Okay, so that means the first one is they are going to give the products which provide a system for intercellular communication, and they are going to recognize very essential antigen by the T cells. So essential for recognition of antigen by the T cells, and they help the immune system in distinguishing, that is self antigens on the non-self, that is its own cells from the foreign cells. And each individual is going to have specific HeLa protein, just like the fingerprint prints. That means the MHC or the HeLa proteins are going to differ from each individual, and it is going to be unique for individual. And the T helper cells with CD4 molecules on their surface antigen is associated with class 2 molecules that we have discussed, and while T cells with CD8 are associated with MHC1 class 1 molecules. And the MHC3 products are important, as I told you, for complement system and also responsible for many cellular events. So these are all the functions of MHC, that is major histocompatibility complex. So until now we have discussed the types of MHC, that is MHC class 1, class 2, class 3, and we have discussed the functions. So let's have a glance of the difference between the MHC class 1 and the MHC class 2. So here is the thing which is going to be present on the surface of all nucleated cells except RBC, I told you. Then it is present on the APC, that is antigen presenting cells like B cells, macrophages, dendritic cells, etc. And these are going to be encoded by HeLa A, B, C genes, and this is going to be coded by HeLa D genes, isn't it? And then we can also having consists of two non-identical chains of alpha and beta, and alpha 3 is the binding co-receptor. Here it is, alpha 3 is the binding co-receptor for T cell, that is CD8, and here beta 2 is going to be the binding site for the T cell co-receptor CD4. And this MHC1 is mainly interacts with the cytotoxic T cells, that is TC cells, and MHC2, that is class 2 molecules, will interact with the T helper cells. Okay, and then coming to alpha chain is encoded on MHC locus of chromosome 6 and beta is 15. Whereas in the class 2, both alpha and beta peptide chains are coded on the same MHC locus, that is chromosome 6. Then the MHC class 1 is mainly responsible for graft rejection and clearance of endogenous antigens, that means internal antigens. Whereas the class 2 is mainly responsible for graft rejection and clearance of exogenous antigens, that means outside the cell, like bacterial toxins, viral toxins, etc. And even that the MHC molecules, the alpha 1 and alpha 2 domains, form the antigen binding pocket, which is not free or closed. So here you can see in the figure, so here it is free. In the MHC, it is going to be of closed, not free. It is closed. But in the MHC class 2, here the antigen binding site is open or free. So these are the few differences between the MHC class 1 and class 2 molecules. So overall in this MHC class 1 and 2, we had seen their differences, their functions, their structure, everything. Okay, so that's all about the major histocompatibility or HeLa, that is human leukocyte antigens complex. I hope so you understood well. Thank you.